Hello, good afternoon to all. My voice is audible. Please let me know. Yes. Good afternoon. Good afternoon, Sudhipati. Yeah, my voice is audible to you. Please let me know. My voice is audible to you as we have decided that we will meet after the lunch. So I am here along with you. Please let me know my voice is audible to all of you. My voice is audible to all of you. Please let me know. Okay. So how are you all? And have you done your homework or not? Please let me know. Have you completed the homework or not? Yesterday we have discussed about interactive genes, about epistasis. So I hope all of you have done it or not. Please let me know whether you have completed the homework or not, because some of you have sent me the photos in the Telegram group regarding your homework. So have you done it or not? Please let me know. So yes, some students have issues regarding the timing of today, which is on Saturdays and Sundays. So these videos are available on YouTube. You can watch it at any time when you will be free, because many of you have some exams or something. So you can watch it later also when you will be free. So no issues regarding it. Okay. So I will share the link with all of you. If you are not able to join the class right now, you will be able to watch it later also. So no worries regarding it. So we have some students like Shreya Rahman and then Soham Banerjee also. So many of you have completed the homework. Deshna, we have done epistasis, that is recessive epistasis and dominant epistasis. But we have also done yesterday additive genes. Some of you have not done yet, no issues. But it's my request or suggestion to all of you that if I gave you some homework, you have to complete it, okay? Because many students have not done it right now. So additive gene effects, you have not given me the example of additive gene effects. Maybe because of yesterday's network issue, you are not able to go through it completely. So I will start the additive genes also today, so that all of you will be able to understand it clearly. So my voice is audible to all of you. Please let me know. My voice is audible to all of you. Please let me know. So we have to do types of gene actions, quantitative traits, qualitative traits, along with multiple alleles. These things we have to complete. Okay. So we have to complete types of gene action, quantitative genes, qualitative genes, and multiple alleles. Then we have something related to mutations, multiple factor hypothesis, cytoplasmic inheritance, gene expression, lack of brain. There are so much different things which are there in your syllabus. Even DNA replication is there. DNA replication, DNA structures, RNA replication, transcription, translation, which is basically central dogma. Okay. So we have to cover all these things. Okay. So have you done these things before also? Any one of you have done genetics before also, or these lack of pain or gene expression? I hope all of you, because these are the terms or these are the simple things which are there in our syllabus before also, or you all are new to the syllabus. Have you done it before also, or you are new to it? Please let me know. Yes, we have to complete the remaining validation. So you have done this before also, or you are new to it? Please let me know. Okay. So some students are completely new to the genetics. Okay. Yes, if some of you are not able to go through today's session, no issues at all. You can watch it anytime when you will be free. So you can watch it any time when you will be free. So I am just waiting for few more students to join. Otherwise, I will start the session. Yes, yes. Good afternoon, Priyasha. How are you? Okay. So all of you, where is your homework? I suggest all of you to please complete the work which I gave you. So yesterday we have discussed about epistasis and we have discussed about, yeah. So we have discussed about epistasis and we have discussed about additive gene effect also. So let me start from here now. So yes, we have to discuss all these gene interactions. We have to study additive gene effects. We have to do epistasis, recessive epistasis, dominant epistasis, complementary genes, duplicate genes, polymeric gene action and inhibitory gene action. So all these are the gene interactions which are there. And one more thing which you have to keep in mind, you have to keep revise or you have to keep in mind the example of these interactions. Like for recessive epistasis, the interaction is the example is coat color in mouse. So you have to keep in mind that in recessive epistasis the ratio is 9 is to 3 is to 4 and the example is coat color in mouse. Similarly for dominant epistasis the ratio is 12 is to 3 is to 1 and the example is food color squash in cooker beta. So the example is fruit color. That means we have white color, yellow color and green color. And the ratio which we get will be 12 is to 3 is to 1 in case of dominant epistasis. In case of recessive epistasis, the ratio is 9 is to 3 is to 4 and the example is coat color in mouse. So the example is coat color in mouse and the ratio is 9 is to 3 is to now we have the ratio is 12:3:1, and we have white color, yellow color and green color. Similarly, in case of recessive epistasis, we have a guthi color, albino color and black color. So you have to keep in mind the examples of it. In case of complementary genes, we have to go with the flower color in Lathyrus, and the ratio will be 9:7. Similarly, in case of duplicate genes, the example is fruit shaped capsula. The ratio will be 15:1. So you have to make the chart of all these gene interactions. Tomorrow I will, or you have to send it in Telegram. I will check this chart also. So in case of duplicate genes, the example is fruit shaped capsula, and the ratio will be 15:1. In case of polymeric gene, in case of polymeric gene, the example is fruit shaped in squash, and the ratio will be 9:6:1. So we will discuss all these interactions in detail. We have already covered recessive epistasis and dominant epistasis. Additive gene effect has also been done by us, but I will revise it once again now. So the additive gene effect, the example is comb shaped chicken. The ratio will be 9:3:3:1. The ratio will remain same as the Mendelian ratio, but yes, there are some differences here, that when we are taking capital R and small p, the color is, the shape of combs is rose. So we have rose comb when we have capital R. In case of small r and small p, we have single comb. In case of only capital P, we have P comb. So we have two varieties, or you can say two crosses or two characters, rose comb and P comb. When we are crossing rose comb and P comb, so instead of getting either rose comb or P comb, we are getting the another trait or another shape, which is walnut. So when the rose comb and P comb are crossed, so instead of getting the either the rose comb or the P comb, we are getting the walnut form. So when capital R and capital P, they both are present, we will able to get the walnut shape or walnut comb. Why? Because here not only one is dominant or one is recessive, rather both are dominant. So capital R and capital P, they both are co-dominant. So they are showing, they are giving us the another comb shape, which is the walnut shape. So here we are able to get the walnut comb in the presence of capital R and capital P. So this was the very first gene interaction, which was described by Watson and Punnett in 1906. So this is the first example of gene interaction when we are crossing two dominant genes and they both are able to express the another character. So instead of giving us rose comb or P comb, we are getting the walnut comb. So inheritance of comb morphology in chicken was described by Watson and Punnett in 1906, and it is the very first example of gene interaction. So when capital R crossed with capital P, we are getting walnut shape. The ratio will remain same as the Mendelian ratio, that is 9:3:3:1. But yes, this is the interaction between the genes. So are you able to get it or not? Please let me know. I just described it here because some of you are not present yesterday, so I have to revise it again. So we are getting, when capital R and capital R is present, we are getting walnut shape. So when only capital R along with small p is present, we are getting rose shape. Small r and capital P gives us P shape, and small r small p gives the single shape, or you can say single comb. So are you able to get it? Please let me know. Yeah, you all will get the slides, no issues at all. I will share it in the group, but are you able to understand it or not? Please let me know, because some students are not there yesterday, so I have to do it again. So are you able to get it or not? Is it clear to you? Because you have not done this interaction in your homework, so you have to give this interaction and you have to show me the table also of all these interactions, in which you have to tell me the ratio, the example of that particular gene interaction. So is it clear to all of you till now? Please let me know. Okay. So now we have done epistasis also. Yesterday we have done epistasis, so I will not discuss it again. Now we will move forward to our another interaction. Like here in this case of dominant epistasis, the example is summer squash, or you can say cucurbita pepo. So you have to remind the gene interaction, the example of that gene interaction, and the ratio which we will get. So we will get 12:3:1. So the ratio is 12:3:1 in case of summer squash or in case of cucurbita pepo. Okay. So we are getting 12 white, 3 yellow and 1 green color. So we are getting it 12 white, 3 yellow and 1 green. So after it we have another gene expression which we have to study. So this is the dominant epistasis in case of fruit color in summer squash or in case of cucurbita pepo. Now we have another gene interaction which we have to study is complementary genes. Complementary genes. Now what is basically complementary genes? The example of this complementary gene is flower color of Lathyrus odoratus, or you can say pea grass. So like you have to keep in mind the example. The example is flower color of Lathyrus odoratus. Now what is basically complementary genes? So two genes are responsible for a particular phenotype. Production of one phenotype requires dominant allele of both genes controlling the character. So I will explain you with example. So let me read it again. Two genes are responsible for a particular phenotype. So if we want to get, suppose we want to get white color. For white color we need two genes, suppose capital W and capital P. So for getting a particular character we require two genes. Production of one phenotype requires dominant allele of both the genes controlling that particular character. So the complementary genes are now present on separate gene loci that interact together to produce dominant phenotypic character. Neither of them, if present alone, can express itself. It means these genes are complementary to each other. So in case of complementary genes, both genes are required together for the expression of a new phenotype or for the expression of a character. So if they are present alone, they can't express itself. Getting my point? So they have to interact together to produce the phenotypic character. If they are present alone, they will not be able to express itself. It means that these genes are complementary to each other. So in case of Lathyrus, basically the color of flower is either purple or white. Purple color is produced. This phenotypic character will be produced only when dominant A is complemented by dominant B. So when we have capital A and capital B, only then we are able to get the purple color. It means the presence of both dominant allele is required, otherwise the color will become white. Let me show you the example. So this was given by Batson and Puneet in case of sweet pea, which is Lathyrus odoratus. So what they tell us, I'll just give you. Okay, so what is their observation? They have demonstrated in sweet pea that purple color of flowers develop as a result of interaction of two genes C and P. So basically two genes C and P are needed. So two genes are needed, C and P, they are needed. In the absence of dominant gene C or P or both, the flowers are white. So in the absence of the genes C or P, the flower color will be white. It is believed that gene C produces an enzyme that catalyzes the formation of necessary raw material for the synthesis of pigment anthocyanin. Hello. ਸਤਿ ਸ਼੍ਰੀ ਅਕਾਲ ਸਰ ਤੁਸੀਂ ਫ੍ਰੀ ਹੋ ਮੈਂ ਤੁਹਾਡੀ ਕਲਾਸ ਲੈ ਰਹੀ ਸੀ ਫਿਰ ਮੈਂ ਆਪਾਂ ਗੱਲ ਕਰਦੇ ਹਾਂ ਇੱਕ ਵਾਰ ਤੇ ਮੈਂ ਤੁਹਾਨੂੰ ਵੀ ਗੱਲ ਕਰਦੀ ਹਾਂ Hello, my voice is audible, please let me know. I'm sorry for the distraction in between. My voice is audible? I think it's audible. My voice is audible? Okay, thank you Nidhi, thank you. So yes, now please try to understand this complementary. It is little bit confusing, but please try to understand. Yeah. So Batson and Puneet have demonstrated that in sweet pea Lathyrus odoratus, purple color of flower develop as a result of interaction of two dominant genes C and P. So when dominant genes C and P are present, the color of the flower will be purple. Getting my point? When the dominant G, capital C and capital P are present, the color of the flower will be purple. Okay. And in the absence of dominant G, C or P or both, the flowers are white. So if we have capital C but small p, the color will be white. If we have small c but capital P, the color will again be white. We have small c, small p, the color will be white. So the purple color will be only there in the presence of capital C and capital P. So it is believed that gene C produces an enzyme that catalyzes the formation of necessary raw material for the synthesis of pigment anthocyanin. So the gene C, it produces an enzyme which is required for the functioning or for the color purple, which is by the anthocyanin pigment. So anthocyanin pigment will only be produced by the enzyme that is produced by gene C. And gene P produces an enzyme which transforms the raw material into pigment. It means that pigment anthocyanin, which gives the purple color, is the product of two biochemical reactions. So the purple color which we will get, it is the product of two biochemical reactions. The end product of one reaction forms the substrate for the other. So substrate B will only be formed by the substrate A. So substrate A will changes into substrate B with the help of gene C, and substrate B will change into anthocyanin with the help of gene P. So both gene C and P are needed for the purple color. Okay. So purple color will only be there if we have capital C and capital P. So only with the help of capital C and capital P we are able to get the purple color. Otherwise the color will be white. So here we are able to get the ratio of 9:7 instead of 9:3:1 of the Mendelian ratio. So purple and white color is there. But purple will only be there if we have capital C and capital P, or you can say capital A and capital B. Otherwise the color will be white. So is it clear to you? So right here we have white flower and white flower. When they are crossed together, when we have capital C and capital P together, we are getting the purple color. Otherwise the color is white. So purple color will only be produced when we have a capital C and capital P. Capital C, capital P. Otherwise the color will be white. So that is why the ratio is 9:7. So this is again the same thing, that from one substrate to another substrate we need gene C. From another substrate to the product anthocyanin or the purple pigment we need gene P. So both enzymes are needed for a purple color. If any one will be missed, suppose this is not there, then purple color will not be there. This reaction is not there, then the purple color will not be there. So both are needed for the purple color. Okay, so both are needed for the purple color. So from the colorless we are able to get the purple color in the presence of two alleles or in the presence of two genes, which is capital C and capital P. So are you able to get it? Please let me know. All of you. Are you able to get it now? Actually, we are not. Are you getting it or not? Diva, are you getting it now or not? Okay. Now let me revise it again. So yeah, this is, you are not getting the substrate from anywhere. This is the observation by the scientists Batson and Puneet. This is the observation by the scientists that how we are able to get the purple color. That there is wide variety, there is again a wide variety. So they are the substrates basically. So how from these two wide varieties we are able to get the purple color. So the scientists, they feel so much they feel so much surprised that how by crossing two white flowers, how by crossing two white flowers we are able to get the purple flower. The focus is on that thing particularly. Then they came to know that this one was the reason of the formation of this gene interaction or the complementary genes, that how we are able to get the purple variety by crossing two white flowers. How we will get this purple color. So that is why when the scientists were working on this flower, they came to know that both of these, that means both of these means that this capital C and this capital P, both of these have some enzymes which when combined together or which when react together, they will give us a purple color. So scientist was able to know that the purple color will only be with the help of a pigment which is anthocyanin, and anthocyanin pigment will only be formed by the presence of both these alleles or by the presence of both these genes. So that is why they came to know that gene C, which is this one, it produces the enzyme which catalyze the formation of raw material for the synthesis of pigment in the flower basically. So substrate and substrate B are basically what? They are the changes which are there, the color change in the flower. So they will able to know that how from the colorless white flowers we are able to get the purple flower. The main reason of complementary gene or the existence of complementary gene is that from the white flowers we are able to get the purple flower. Then they, by doing the experiments, they came to know that the purple pigment is because of anthocyanin, and anthocyanin will be only produced in the flower with the help of with the help of both capital C and capital P. So that is why they are known as complementary genes. What do you mean by complementary? Complementary is basically when they are complementing each other. So complement basically when they are complement, when we are giving compliment to someone. So that means when they both are knitted together, the capital C and capital C both are knitted together for the color, for the purple color in case of flowers. So both are required together. They are complementing each other. Okay, so they both are complementing each other for the formation of a, or for the existence of a purple color in the case of sweet pea flower. So is it clear to all of you? Is it clear to all of you? Please let me know. John, is it clear to all of you? Deepshika, Diva, you have to keep this thing in only in mind that purple color will be only produced by the presence of both dominant alleles, which is capital C and capital P. Got it? Okay. So now, like we have the complementary genes, we have another gene which is supplementary gene. Let me check it whether we have it in this presentation or not. Otherwise, I will let you know again. So after the complementary genes, we have another genes which are known as duplicate genes. So, okay. So we have another genes which are known as duplicate genes. Now what are basically the duplicate genes? Let me share the PPT. So yes, what is basically duplicate genes? From the name obviously we are able to know that they are the duplicate of the original one, or you can say the original one. So now what happens in the duplicate gene? If the dominant allele of a two gene loci, whether inherited together or separately, the ratio 9 is to 3 is to 1 gets modified into 15 is to 1. So the example of it is capsules of shepherd's purse capsula. So this is the example. We have shepherd's purse, the example of capsula. In the capsule of that, this type of interaction occurs. Now what happens here? Capsules occur in two forms. They are either triangular or they are either top shape. Let me show you like here. So they are either triangular, the capsule, or top shape, or you can say heart shape or and narrow shape. Okay. So they occur in two shapes, triangular and top shape. So when a plant with triangular capsule, when a plant with when a plant with triangular capsule is crossed with one having top-shaped capsule, in F1 only triangular characters appear. Okay. So when triangular character is mixed with the narrow character, only triangular character appears. The F1 offspring by crossing over produced the F2 generation with triangular and top-shaped capsules in the ratio of 15 is to 1. So we will able to get triangular only, and only where there is small a and small b we are able to get the narrow shaped capsules. So here the ratio is 15 is to 1. Hello? नहीं, मैं क्लास में थी. अरे तो मैं तो सर कल की बात हुई है, इन्होंने बच्चों ने कहा था कि हमारा जल्दी ले लो लेक्चर. चलो फिर सर उनको कहो ना कि वीकेंड्स पे ही आगे पीछे हुआ, अदरवाइज उसी टाइम पे क्लास लेती हूँ. नहीं चलो फिर कल उनको कहना कि कल को. चलो मैं देखो, ठीक है, चलो मैं डाल दूँ, रुको. ओके 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 मैं भी देख लेती हूँ. ओके ओके तुम्हारी बात। ठीक है ठीक है ओके ओके। हेलो, so my voice is audible to all of you. I'm really sorry to be to have to. Hello, my voice is audible to all of you. Please let me know. Pooja, sorry, Nidhi, Bhavna, Bhavni, my voice is audible. Please let me know. All of you. Hello, hello, Nidhi. All of you, my voice is audible. Please let me know. Hello, my voice is audible to all of you. Please let me know. Hello, my voice is audible. Hello, please reply. Any one of you, please reply if possible. Yes, some of you have some issue regarding the timetable or the class which we have taken right now, because they are just asking the authorities that ma'am have taken the class just after the lunch. So I already informed you before also that we will take the class after the lunch. So what is the problem in it? So students have some problem regarding the lecture, that ma'am is taking the lecture. But yeah, but I already informed you before it that I will take the class a little bit early. Definitely on Sunday I will take the lecture. Little bit early. So I don't know why they are creating so much of uh, problem now. Are you able to listen to me? Any one of you? Have you any problem regarding the timings, because they are messaging in the group? So are you able to listen to me? Please let me know. Have you any problem regarding the timetable now? Because I have taken it, because you all know that this is Sunday. And I already informed you before also that we take the special class of 2 hours, and we have to take it little bit early. So why the students are creating so much of nuisance right now? I don't know. So if you are able to listen to me, please reply because I am not able to get you. Are you able to listen to me or not? Okay, it was you, Priyasha, who just messaged the higher authority. It was you, Priyasha. Please reply if possible. Priyasha, please reply. You are the one who messaged right now. So I have to stop my class in between because of all these troubles. Now my students are waiting for me from so much long time, and we are discussing so much good things related to genetics. And what's your problem? I'm not able to understand. Priyasha, please explain it properly. What's your problem right now? What's your query? Please let me know. Otherwise, I have my own students to which I have to teach the genetics. So instead of just focusing on all these things, I am not able to. If you want to say something, type it right now. Otherwise, you are just messaging to the higher authority. What's your problem? You have to discuss it in a Telegram group or also along with me. So please reply. What's your problem? Otherwise, I have to complete the gene interaction. Some query, please let me know. If you have some query, please let me know. Priyasha Garg, please speak up now. Please speak up now, Priyasha. No, just Divya, just wait for once a minute only because these guys are creating so much of problem right now. I am just focusing on the topic. We are just doing it very nicely and smoothly. But I don't know what happened to them. They suddenly message to the higher authority. What is your problem, Priyasha? Please speak up now. I am just waiting for your reply. Priyasha Garg, are you able to listen to me? Please type your message as soon as possible. Priyasha, now they will not reply. Priyasha, बच्चे, what is your problem? Please let me know. I have my students waiting for me. I have to complete the lecture. If you have any query, you can ask me at any time. You can even message me personally. But what is this type of behavior? You are not discussing anything in the group and just you can say you are just messaging to the authorities. What is your query? I am just asking you. Please let me know, Priyasha Garg. What is your query? I am waiting for your answer. My whole class is waiting for your answer right now. Please let me know. We all are waiting for your answer. Otherwise, I have to complete the duplicate genes. I have to complete the gene interactions. What is your question or query? We are waiting. Please let me know. Yeah, please let me know. Yes, please. We all are waiting. Please let me know if you have any query. Please, let me know. We all are waiting. So all of you, when you will have your next class, your proteomics class, when will you have this? By 5 you have your next class, so we have 1 hour more. So please let me know when you have your next class. Okay. So please let me know when you have your next class. Some of you has messaged right now that in the class only 19 students and ma'am is just taking the class. So those 19 students which are there, they are punctual, they are on time. and they are waiting for me from the morning, so I have to take the class. What is wrong in it? Some of you from your class right now message to the authority that ma'am begun the lecture by 3:15, and there are only 13 students present in the class now. The others are not aware that it started. So you are aware now that class is started. So instead of focusing on others, you have to there in the class. You have to listen to what the teacher is saying. If my 19 students are there along with me, I have to focus on these 19 or 20 students because they are able to listen to me. They are waiting for me, and I have to deliver the lecture to the sincere students. You are here, you are live, and rather than focusing on the topic, you are just concerning about other students. That many of the other students are not aware of it. You are aware. You have to listen to the lecture. Then, if you are aware, instead of thinking of anyone else, why you are thinking of anyone else? Just think of your own self. That ma'am is delivering the lecture. If you are interested in listening to the lecture, then most welcome. Otherwise, you can watch it later on also. Urja Joshi, you are not the only one who gets disturbed. The trainer who is teaching you, the trainer who is getting, just taking the notes and just delivering the lecture, when he or she will get the call immediately, then of course the trainer will definitely get disturbed also. So anyways, we have to complete our avatar there in the duplicate genes. So in case of duplicate genes, the example is basically the example is basically Shepherd's purse. Now what happened in here, we have two genes, we have gene A and gene B. So they are of two types. Either the shape of these fruit will be triangular, that means the shape will be like this way. It will be like this way, the triangular shape, or the shape will be narrow shape. So it may be narrow shape. So what happens basically when we are crossing the capital A and capital B with small a and small b, in the F1 generation we are getting triangular shape. That means capital A and capital B. Getting my point? So we are able to get, let me show you again. Yeah, so we are able to get capital A and capital B, which is triangular. So anywhere if we have any of the character, either capital E or capital B, the ratio will be, or the shape will be triangular. So when they both are present together, the shape is triangular. When they are not present together, rather they are present singly, that means either capital A is present or capital B is present, then the shape will be triangular. If A and B are both present together, again it is triangular. When only small a and small b is present, then it is another shape, which is the top shape. So the ratio is here 15:1. So we have 15 triangular and we have only one, which is the recessive form. We have only one top shape. So this is an example of gene interaction. Two genes involved in the same pathway. It is based on the idea that some genes may be present more than once in the genome. So when capital A is present, the shape is triangular. Okay. When capital B is present, again the shape is triangular. When both A and B is present, again the shape is triangular. And we have only when they are present in recessive form, small a and small b, then the shape is top shape. So getting my point? So either A or either B we will get heart shape. Only in the presence of small a and small b we are getting the narrow shape. So this is the fruit shape in case of Shepherd's purse, and this is known as duplicate genes when both are able to express itself. So in a cross between two lines differing in fruit shape, heart shape versus narrow, F1 generation shows all heart shape like intragenic dominance, but F2 generation shows a ratio of 15:1 not 3:1 as that of monohybrid cross. It means that the trait fruit shape is controlled by two genes A and B and their alleles, which is small a and small b. So 15:1 ratio is there instead of the Mendelian ratio of 9:3:3:1. So the triangular shape results by the presence of at least one dominant allele of either gene. The two genes appear to be identical in function and is in contrast with the complementary genes. Okay, so duplicate genes provide alternative genetic determination of a specific phenotype. So we are able to get triangular shape either with the presence of TV together or capital T or capital V. So we are able to get the triangular shape. So this is known as the duplicate gene action of epistasis, or you can say duplicate genes, when we are able to ratio of 15:1. So is it clear to all of you? Similarly, we have one more example of this duplicate genes, which is in case of petal color in snapdragon. So in the snapdragon, the flower is generally red in color, and only it becomes white in the absence or in the presence of recessive genes. So whenever a dominant gene is present, the trait is expressed. One allele is sufficient to produce the pigment. So here two alleles are not required as in case of complementary genes. Rather only one allele, either capital C or capital P, only one is needed to produce the particular pigment for the red color. Like in case of complementary genes, we need both C and P for the purple color, but here in this we only need one gene, either A or B, for the production of red color. So is it clear to all of you? Please let me know whether this you can say this duplicate genes, are you able to get it or not? Are you able to understand how it is different from the complementary genes? Are you able to understand duplicate genes and what is the difference between duplicate genes and the complementary genes? In case of the duplicate genes, we have that both genes are not needed together for the production of a red color. Like in case of complementary genes, both capital C and capital P is needed for the production of the purple pigment or purple color. So is it clear to all of you? Please let me know. Is it clear to all of you? Please let me know. Is it clear to all of you? All of you are able to get it or not? Please let me know. Okay. Okay, good. So is it clear to all of you? Please let me know. All of you, are you able to understand duplicate genes? Are you able to understand complementary genes? All of you, please let me know. Okay. So now we have another gene which we have to discuss. We have another gene. Okay. So it's okay. So now we have another genes which are known as, we are just discussing about the gene interactions which are there. So we are just discussing the gene interactions. Now we have another gene interaction which is known as polymeric gene interaction. We have another gene interaction which is known as polymeric gene interaction. No, these traits are not only shown by these plants only, but the scientists have observed these interactions in only these plants. Like in sweet pea, Mendel has shown the interaction in case of sweet pea. Similarly, Batchen and Puneet has seen the interaction in case of Lathyrus. So they have shown the interactions only. So it is only the work which is done by the scientist on a specific single plant. Okay, so now we have another interaction which is polymeric gene action. Now what happened in case of polymeric gene action? The ratio is 9:6:1. In polymeric gene action, the ratio is 9:6:1. Now what happens here? Two completely dominant genes controlling a character produce same phenotype. So we have two dominant genes like capital A and capital B controlling a character produce same phenotype when the dominant gene alleles are alone. But when the dominant alleles are together, the phenotypic effect is enhanced and becomes more cumulative or additive effect. So they are able to show the effect alone also, but when they are present together, the effect is more enhanced and becomes cumulative or additive effect also. So the examples are on length on barley fruit or fruit shape of squash. So either we will get the shape disc, circular or long. The ratio is 9:6:1. So what is basically happen here? Now this is the example in case of length of barley fruit. In case of barley, so what happened when capital A and capital B are present together, it will be long on. When small a and small b present, the shape will be, the length of the barley fruit will be on less. But when both A and B are present together, the shape is long on. When capital A and capital B crossed with small a and small b, we are able to get long on in the first generation. So we are able to get long on in the first generation, and we will get the medium shape. So we have long shape, we have small shape, and the medium shape will only be there when we have only a single dominant allele. So when we have the recessive forms, that means everything is in small, then we will get on less. So only one is on less, otherwise when both A and B are present, the shape will be long. When small a and capital B is present, the shape is median. Capital A, small b is present, shape is again median. And when small a and small b is present, the shape is onless. So this is the example in case of polymeric gene action. When the both are present, the shape is long. Both are absent, the shape is onless. But when any one of the dominant is present, the shape is median. Similarly, in case of this squash, fruit shape in squash, when both are present, the shape is disc. When both are absent, the shape is long. And when either one is present, one is absent, the shape is circular. So instead of getting two traits only like tall and draft, purple and red and white, round or restricted, we are getting three shapes. So instead of getting two shapes, we are able to get here, three shapes. Okay, getting my point? So instead of getting the two traits only, either tall, either short, or white or red color, we are getting three types of color or three types of shapes. Are you able to get it? Then after it we have another interaction which is known as inhibitory gene action. We have inhibitory gene action. So what is basically inhibitory gene action? The inhibitory gene action, the example is in maize color and the ratio will be here 13 is to 3. So in case of inhibitory gene action, the ratio will be 13 is to 3. Now what happens in this inhibitory gene action? One dominant gene produces concerned phenotype and its recessive allele produces contrasting phenotype. So we are able to get two types of phenotypes. The second dominant has no effect on the concerned phenotype but stops expression of dominant allele of first gene. So when both dominant alleles are present, phenotype as that of the recessive homozygote is produced. So basically what happens here, when capital R and capital I are present, the shape is white illuron. In the presence of small r and small I, again the shape is white illuron. So what will happen basically here, let me show you. When capital R and capital I is crossed with small r and small I, we are getting the white color. Getting my point? So we will get white color, white color, white color when capital R and capital I is present. But there are some cases when we are getting the red color. There are some cases when we are getting the red color, and this red color is only present in the presence of small I. So when we have capital R along with small I, we are getting red color. So when we have capital R along with small I, we are getting the red color. So the ratio here is 13 is to 3. So 13 will be remain white, but only when we have capital R and small I, the color will be red. Or this is another example of inhibitory gene. The example is feather color in fowl. So similarly here, this is also the inhibitory gene where one gene inhibits the expression of another gene. So here what happens? One gene is not allowing the expression of the other gene. So if you will be able to see here, when both capital A and capital B present, the color is white. When both capital A and B present, the color is white. When only one A and small b is present, again the color is white. When small a and small b is present, again the color is white. Now here the color becomes changed in the presence of one small a and another capital B. So what happens here now? In the presence of small a and the capital B, now the color is red. So what happens here? B is responsible for color. So this capital B is responsible for color, but in the presence of A. So this B is responsible for red color. Okay, but in the presence of capital A, this allele is not able to express itself. So this allele will only express itself in the presence of small a. Getting my point? So this capital A inhibits the another allele which is B. So when A and B are present, the color is white. The color becomes red only in the absence of small a and in the presence of capital B, because capital A inhibits its color. Are you able to get me? Please let me check. Are you able to understand inhibitory genes or not? Please let me know. Are you able to get it or not? Please let me know. Are you able to understand inhibitory gene? All of you, please let me know. All of you, please let me know. Are you able to understand it? Okay. So you are able to get the meaning of inhibitory gene. What is the ratio in case of inhibitory gene? And what is the example in case of inhibitory gene? What is the ratio in case of inhibitory gene? Please tell me what is the ratio of inhibitory gene. All of you, what is the ratio in case of inhibitory gene? What is the ratio of inhibitory gene? Okay, okay. So the ratio is 13:3 because capital B will only show its effect in the absence of capital A. So it will show its effect only in the presence of small a. So up to now, is it clear to all of you? Yes, yes, very good all of you. So up to now, is it clear to all of you? Up to now, is it clear to all of you? So I hope you all will get it. So this is the whole things which are there in the gene interaction. I will share the PPT with all of you right now after this lecture. I will share the PPT. So you have to go by your own self. Similarly, one masking gene action is there in which the ratio is 12:3:1. It is similar to again to our previous gene interaction. Kindly go through it by their own self. So when we have capital B and capital Y, the color is black. So you have to go, you have to read it by your own self. So when we have small capital and capital, the color is black. When we have small b, small y, the color is white. Now one dominant, one recessive, the color is black. One dominant, one recessive, the color is yellow. So instead of getting two colors only, here we are getting the three colors, and the ratio is 12:3:1. So kindly go through it by your own self. I will share the PPT. So this is the only thing which we have to discuss in case of gene interactions. So is it clear to all of you? Now we have to move further to our next topic, which is there in your syllabus, which is quantitative and qualitative traits. So shall we start now? Please let me know. Shall we start now? Okay. So now I'm just going to start another topic which we have to cover. The very small topic, so I will cover it very fast. No worries. I will start it now, and we will try to complete it. Do you have any another class right after this lecture series? Do you have any another class also? Okay, okay. So we will try to compile it by 5 p.m. So let's start. The topic is very small, so I want to do it today. Okay. So please cooperate, all of you. Yeah. So we will do quantitative characters now. It's a very small topic, so that is why I think we will cover it today. Okay. So what is quantitative characters? We have to study this. The topic is very small, so I will try to complete it fast. So yes, what is basically quantitative characters? Quantitative genetics, or you can say quantitative characters, have the possibility of continuous variations. So it was in early 70s, 60s when one scientist reported first case of continuous variation due to quantitative trait. So in quantitative, we have two terms, quantitative and qualitative. In case of quantitative trait, continuous variation is there. In case of quantitative traits, continuous variation is there. So quantitative characters are an inherited characters. What are basically quantitative characters? They are an inherited character that expressed phenotypically in all degrees of variation between one often indefinite extreme and another, a character determined by polygenes. Yeah. So what happens in quantitative characters? All are expressed phenotypically in all degrees of variation between one. So let me show you the example, then you will able to get it. We have two terms. We have qualitative, we have quantitative. In case of qualitative, when we are working on a qualitative, it is under the control of single gene. The qualitative is under the control of single gene, but the quantitative, they are under the control of more than one genes. So right here, if you are able to see, the inherited character that expressed phenotypically in all degrees of variation between one often definite extreme or another, a character determined by polygenes. So the quantitative character is controlled by more than one gene. Like in case of Mendel ratio, one character is controlled by one gene. But here it is opposite. Here one character is controlled by more than one gene. So in case of quantitative trait, one character is controlled by more than one gene. So character of a kind, this continuous variation, distinct phenotypic class. In case of quantitative traits, characters of degree, here is the variation is continuously, like here the variation is discontinuous, here the variation is continuously phenotypic measurement from a spectrum. So it concerned with individuals mating and their progeny. The quantitative traits are quite important. Why? Because they are concerned with the population of organism. So these quantitative traits concerned with the population of organism consisting of all possible kinds of matings. So these quantitative traits, they are quite important because they are continuous variation, they have the effect of multiple genes, and they are just concerned with the whole population instead of a single individual. So here we are able to get the statistical data also in case of quantitative traits. So in the case of quantitative traits, the common examples are skin color. So we have not have a black and white skin color, rather we have different types of skin color, or you can say eye color. Like we have bluish eyes, greenish eyes, brownish eyes. So we have different types of eye color, hair color, weight, height. All these are controlled by polygenes. All these are controlled by polygenes. Here, the form, the round or wrinkled seeds of pea, they are qualitative trait. That means single gene controlling the single character. Here in quantitative trait, seed color in wheat, ear size in maize, corolla length in tobacco. All these are quantitative traits. Quantitative traits. It means they are controlled by many genes together. So similarly, in animal, size in chicken, size in rabbit, all these are the quantitative traits. So the very common example of quantitative trait is skin color in humans or eye color. Getting my point? We have two types of traits, or we have two types of character. We have qualitative character, we have quantitative character. So in the case of quantitative character, we will get skin color, eye color, hair color. So it is because of the polygenic control, it is the continuous variation and it concerned with the whole population. So skin color is not associated with only a single person, rather it is associated with the whole population. So getting my point, are you able to understand what is the difference between qualitative and quantitative traits? After it, we have the multiple factor hypothesis. Now what happens here? So a gene individually producing a slight effect on the phenotype, but in the combination with a few or many other genes controls as quantitative character is called polygenes. So when more than two genes are controlling a character, it is known as polygenes. Getting my point? When more than two genes controlling a character is known as polygenes. Since there are many genes of this kind for quantitative character, they are also known as multiple factors. Each factor having too small an effect to be traced. So basically what happened? When we are discussing about a quantitative character, it is controlled by many genes, also known as polygenes, and this effect is known as multiple factor. So those genes or alleles which contribute to the trait involved, they are called contributing genes, and those genes which are not involved in this particular character, they are known as non-effective or non-genes alleles. So these are the examples of some quantitative traits like wheat, the color in wheat, skin color in humans, eye color in human. All these quantitative traits are studied very briefly. So if you are able to see in the case of wheat, instead of getting red wheat or white, we are able to get so many variation in between. So instead of getting red color properly or white color, no, we are getting so many variations in between. Similarly in case of humans also. So we are getting red color, we are getting white color. But in between we are getting medium red, very light red. So we are getting so many variations. Similarly in case of skin color. So we are not only black and we are not only white. Rather there are so many in between colors also. So when we are doing the two pair of genes A and B causes a difference in skin pigmentation between Negro and the Caucasian people. So we are not getting only the if black and white they are crossed together, instead of getting black or white we will get the intermediate color. So we will get negro color, dark color, mulatto color, light color, white color. So instead of getting one color or two color, or you can say instead of getting one character or two character, we are getting so many different characters. So we are getting albino also, fair also, mulatto also, dark and negro. So instead of getting a single color, we are getting albino, negro, all are there. So polygenic inheritance of skin color in humans is quite important. Similarly, eye color is also prominent. The eye color may be blue, maybe medium blue, dark blue, grey, green, light brown. So the color may be, the color in the eye is also different. So instead of focusing on a two genes only, they are controlled by many genes. Getting my point? So is it clear to all of you? Is it clear to all of you that how we are able to get different types of colors in case of humans, in case of eye color? Are you able to get it or not? Yes, before discussing about continuous and discontinuous variation, you must know what is basically variation is. What is basically variation? Please let me know, Bhavni. What is variation basically? What is variation? Basically, variation is the change. Getting my point? Variation is the change in the gene level, or you can say a mutation. You can also say it is a mutation. That first we are getting this, now because of some variation or some changes or some mutations, instead of getting this flower, we are getting little bit of this flower, or you can say little bit of this color. Variation is basically the change or the mutation. So there are two types of variation which is continuously occurring, like the skin color. The variation is continuously from so many years back, and this continuous variation is which will stop at some time and then it will start again. So continuous variation is occurring continuously. So the change in the skin color is from the many times years back we are able to get the dark color, and then there are so much variation. Variation means changes, and if it is continuous means The variation is keep same as it was before also. So this is only continuous variation and the discontinuous. We have continuous changes which are occurring, and we have some changes which are for only a specific period of time. Then these changes will not occur again. But in continuous variation, we are able to get those changes again and again. Getting it? So are you able to understand quantitative, the difference between qualitative and quantitative? Yes or no? It's a very small topic, so that is why I decided to discuss it today also. Is it fine? Are you get it or not? Okay, good. So very good. So that is the main thing which we have to discuss today. Now please discuss with me what is the status of your review paper. Have you selected a topic? If you have selected a topic, please share here along with me. Have you selected a topic? Please share with me if you have decided, if you have selected a topic. Yeah, please all of you share your topics which you have decided. Okay, okay, okay. Very good, Pooja. All of you please share, and I will suggest you to keep the title. The title should be quite long, at least of two to four words, so that it will look much attractive. So please share the title, the topic which you have decided. Please share the topic. Anemia caused by genetic disorder. Very good, Bhavni. Very good. Keep it up. Any another topic? Any one of you? Cloning. So don't write only cloning. Make it. The topic should be at least of 4 to 5 words at least. Okay, Priyasha. CRISPR technology for gene silencing. Okay, very good, Priyasha. So, Diva, I will suggest that please make the title little bit longer. Okay, okay, okay. So, Nidhi, what's your topic? Please let me know what's the title of your topic. Nidhi? Okay, no issues, Urja, no issues at all. Take your time. Just focus on your exam right now. No worries. But at least try to select a topic, because I am telling all of you from the very first day that you have to select the topic as soon as possible. Take your time, but you have to select it. Okay, okay, okay. So Priyasha, you have to write at least 5 to 6 pages. All of you have to write at least 5 to 6 pages. Yeah, yeah. Nidhi is very right. Variation is a slow change in the at the gene level basically. Okay, Vishnu, Vish, Vishnavi, Vaishnavi, genetics and Parkinson's disease. Yeah, very good, very good. Please, Vaishnavi, please keep it up. Please write the abstract as soon as possible. Okay. Biofertilizers. Okay. Good. Please keep it up. Please try to write the abstract now. Today is Sunday. You all are free, so you have to write little bit about the abstracts. Yes, Pooja. We will take it on 8 p.m. because I think you are the one who messaged, Nana. Pooja, you messaged. Please reply. Have you any exam or something? Please reply, dear. Have you any exams or something? We will have to we will have to keep it at 8 p.m. because students are not ready to cooperate. So we will try to keep it at 8 p.m. Actually today I take it on this time because I want to do something more. I want to complete multiple alleles also. So that is why I think if few students will not be here, they will watch it later on the YouTube link which I will provide, which you will share obviously, definitely. But the students are not try to cooperate. Yeah. No issues at all. Yes, you can submit it later also. No worries. No worries. Role of bioinoculums in enhancing soil health. Yes, very good, Nidhi. Your topic is very good. Please try to start it as soon as possible. Human genetics. No, the references depends on the type of paper you will read. The content which is there in your paper, how many you have taken it from another paper, the number of references will depend upon that particularly. And Komal, please make your title little bit longer. Instead of human genetics only, make it more attractive. Human genetics. What you want to say in the review paper, it should be implemented in your title. Okay. Genetics and its impacts on human diseases. Yeah, very good. You can keep it. Actually, good. Mm, but Komal, you have to increase the words in your title. Medicinal plants of Sikkim Himalayas as antimicrobial agents. Yeah, the topic is very nice, Lakpa. And from where you belongs to? May I allow to know from where you belongs to? Okay, so you are from Sikkim. Okay, very good. Your topic is very good. Medicinal plants of Sikkim, Himalaya as an antimicrobial agent. Yeah, yeah. Please, please. I'm waiting for your abstract and your review paper. CRISPR technology using in endometrial cancer. But before you write some another title. But this title is also good, so you can keep it. No issues at all. So where is Komal? Komal, please try to make your title little bit long. Okay. So I hope you all will get it, and I hope you all will work very hard. You are here along with me from the past 1.5 hours. That means you are concerned, you are serious about your studies, about your lectures. Otherwise, the rest of the students, they are not even bothered to attend it even for 10 minutes or for 5 minutes or for 15 minutes. I think that much time we all have. We can at least attend the session for 20 minutes, for 25 minutes, but they are not here. No issues, we will complete our syllabus if we have to. So I think that's all for today. We will meet tomorrow on 8 p.m. In the meantime, if the students will have some queries, those students who didn't take the lecture right now, so it's my request to all of you to kindly answer those queries and kindly share the link again and again if they want something, the recordings or something. Please share the link and also tell them the homework which you have to do. So you have to make the graph or the diagram for the interaction of genes or the types of interactions which we have studied. And yes, one more thing which you have to do. I just told you about complementary genes, but right now I didn't tell you anything about supplementary genes. So this is your two homework. You have to make the table or the chart for all the gene interactions, and you have to let me know what is supplementary genes. So you have to tell me what is supplementary genes. So Nidhi, you have to send me your abstract on my email ID. I am just sharing here. So it's my request to all of you, if the students will ask question and I am not able to reply, then you have to answer those questions. Okay, they will ask you something regarding the homework or something, so you have to let them know. So you have to share the syllabus as well as this YouTube link with all the students, so that they will be allowed to watch it again. Okay, when they will be free. So that's all from my side. Take care of yourself. Enjoy your Sunday, and we will meet tomorrow on 8 p.m. And thank you for being here with me. Thank you all. Okay, bye bye. Take care of all. Bye bye. Bye bye all. So I am just now going offline. Take care of yourself. Bye bye. So tomorrow we will do multiple alleles. I want to cover it today also. Okay, we will do it tomorrow. Okay, bye bye.